In 2006, the NICE, the National Institute for Clinical Excellence, refused to fund a £20,000 cancer drug, even though it was available in some parts of the world. Was this justified? Should drug companies be forced to lower the cost of their drugs? A lot of people would instinctively say yes, of course, but we need to be aware of the cost of drug development and the fact that so many drugs fail to get to the end of development causes the cost of those that do succeed to go up. So why then do so many drugs fail? That's what we're going to look at in this short lecture here. The reason why drugs often fail is that the criteria for their success are so strict. For a drug to be marketed, it must be effective, it has to treat the condition with good pharmacokinetics. It has, of course, to be safe, that is, a large therapeutic window and with tolerable unwanted side effects. Of course, all drugs have side effects, but the question is how tolerable they are. It has to be usable, a feasible dosing regime, easy to administer, and of course it has to be economical. So to understand why it's so difficult to satisfy all these, let's have a look at the drug discovery pipeline. This is a standard sequence for drug discovery and development. Here's an outline of the drug discovery development pipeline. Drug discovery, preclinical development, clinical development, regulatory approval, and finally, phase 4. Let's have a look at the first section, which is target selection. You have to start off with choosing a place for the drug to act, and one way of doing that is to use our biological knowledge. So, for example, we know that increases in angiotensin II cause increased blood pressure. So if we want to target blood pressure, we could choose AT1 receptors as your target and develop antagonists. We know that beta-1 adrenergic receptors increase the rate and force of cardiac contraction. So they could be an antagonist, could be a target, it could target beta-1 receptors. We know that angiotensin converting enzyme stimulates the conversion of angiotensin 1 to angiotensin 2. So ACE could be a target, and we could then use things like peptidases. This could be augmented when we know, as we do now, we know now so much more about the genomics and interacting proteins. And once we've got one target, using interactome, we can select a number of other targets as well. It can also be helped by computer modeling, in which case we actually make physical models of receptor proteins, and then we can design drugs which act at the active site or the binding site, ligand binding site of these receptors. So once you've got a target, you then have to find drugs which act on that target, and that's called lead finding. And typically you would create an assay which captures the action of your target and then produces a nice readable hit in a large throughput screen. Which chemicals could you start using to look for? You can use natural products such as artemisinin, which you can see growing in the background here, or there are commercially or even freely available kits consisting of hundreds of thousands of different molecules, and drug companies typically have their own libraries of millions and millions of compounds. Alternatively to finding a target, you can bypass that and simply build an animal model, such as this C. elegans model of spinal muscular atrophy, and look for drugs which rescue the phenotype. Once you've found a molecule which works or a hit, you then have to optimize it. And what you will want to do then is to find the precise details of the molecule which make it effective and alter it until you find one which is even more effective. You then have to pharmacologically profile all your new hits. Hopefully one of those hits is going to result in a drug. You need to know what other targets it reaches. And as you can see, this sort of profiling can find some very dangerous off-target effects. So for example, if you look on this graph here, you can see just how many drugs have dangerous off-target effects. So far we've got something like a 50% fail rate already, and we haven't even got to preclinical development. And in preclinical development, we ask the question, is this drug safe to test on people? We look at the pharmacokinetics, the toxicology, the formulation, is it going to be a cream or a tablet or a powder, and how we're going to scale up the synthesis. We've now got another 50% attrition rate. So at this point again, half of the drugs you've got left will have not passed one of these tests. And this is typical of the press release you get when they have to announce that a drug has failed at this point. We now have a transition from a culture of laboratory experiments to more accountable boardroom style of governance. And from this point on, during the clinical development, we leave the laboratory environment and now enter into the regulatory world, which is governed by documents like this one, Good Clinical Practice for Laboratories. And that takes us into phase 1, 2 and 3 of clinical development. And each of these is designed to scale up the number of patients that you tested on and the kind of problems that you're looking for, such as long-term toxicology rather than short-term toxicology. New technologies are working in our favor, having said all that. So techniques such as machine learning, which look at targets and then predict what sort of ligands should interact at the target, proving successful. And we've recently had halicin, which was shown to be one of the first new antibiotics discovered for a very long time. So what you see from this then is that there are many hurdles that the drug has to go through before it finally gets to regulatory approval.